Good morning, everyone. Thanks a lot to One Nucleus for all the organisation and for the invitation, and a particular thank you for holding the conference 400 yards from my desk, which is quite helpful. So what I'd like to talk about is particularly with relation to in vivo platforms, but particularly talking about the diversity of antibodies. The creation of diversity is obviously an important thing in finding that rare antibody candidate that's going to take you forward. So we've come a bit of a way since the first use of immunotherapy, and in fact this is sometimes people still think this is how it works, because I did a presentation recently to a business awards group who are award of lay people, and one of the questions at the end of the presentation was, well, how many mice will you actually need to supply the world demand for your particular antibody? So I think some people still think you just put a tap on the side of the animal. But obviously things have moved on since Behring's pioneering work. As Mike said in the introduction, so I won't dwell on this, we've had a gradual evolution of what we're doing, as well as the phage display in vitro platforms, rodent antibodies. We've also got a lot of people now innovating with finding antibodies directly from human B cells in patients, particularly in infectious disease. And then the area I'm going to kind of focus on is this aspect of mice with humanized immune systems, because that's what Kymab does, but it's also, I think, quite important when we consider the fully human antibody aspect of monoclonals. Obviously, mice, in principle, with fully human immune systems, we all know how to immunize mice, so if you do this correctly, simply immunizing the mice and then finding human antibodies rather than rodent antibodies is really the ideal target. And clearly one of the big advantages here is that if it works properly, and this is an if, if your transgenic platform works properly, you let the mice do the work, selecting the in vivo antibody, and also doing all the affinity maturation for you. Whilst the mouse doesn't have a fermenter and an impeller, so we can't actually tell you that the antibodies are ready to go to the clinic, but a lot of the biological compatibility will be taken care of in the mice. So in terms of fully human antibodies, which is I think the growing area, in vitro and in vivo platforms have been used about since the same amount of time. As Mike mentioned, of the 9 approved fully human antibodies, 7 are from transgenic mice, 2 are from phage display. And I think that probably says something about the compatibility with biological systems. The less engineering you have to do, perhaps the better. In terms of the diversity of the immune response, finding that candidate that's really going to take the market and be best in class or first in class is actually quite a rare thing. So you actually have to look at a lot of candidates to really get to where you want to be, and therefore the more diverse the starting pool, the better. As far as its in vivo is concerned, there's a number of areas where you can look at intervening here. First of all, the gene repertoire, you want to make that as diverse as possible, which is one of the aspects Kymab is very much taken into account. And make sure that the in vivo system is working correctly in terms of extensive somatic hypermutation and antibody recombination. Obviously, sound biophysical properties are a bit of a given. But really, a key thing which is often neglected is optimizing the sampling. I mean, we did a quick calculation looking at either a human being or a mouse with a full human repertoire. And theoretically, the diversity is something in the range of 4 times 10 to the 14. Which is, an incredibly large number. The trouble is, and particularly if you're using something like hybridomas, how much of that are you actually leaving in the mouse and not even knowing exists? So that is quite a compelling issue. And as far as the Chi mouse is concerned, which is not surprisingly the mouse that we're making from Kymab, and it did initially start with chimeric antibody with CHI, but we thought that everyone would call it Chymab, so we changed it to Chi mouse. The key design principle is to really couple the entire human gene repertoire that exists in a initially single human, but potentially a population of humans. And in fact, we've taken a few tricks to actually widen that beyond the diversity of a human, and really couple that with really what a fully functional murine immune response, so that you really get the best of both worlds. This is a Chi mouse in cartoon form. They are actually white. So the simple aspect is to put all of the variable genes for all three chains, the heavy, kappa, and lambda. We learned from the experiences of Medarex and Abgenix that you want to use a mouse constant region because that will get you optimum signaling in a mouse B cell, and therefore you overcome some of the issues with previous generations. And this is what we call our HKL, which is all three, but we do have a range of strains that we're creating. And really the principle, the whole repertoire, all three loci, and engineered into the endogenous loci, so we're not doing any random insertions or like some of the other technologies that have been around. And we think this is actually important when you get down to the details of looking at how do the antibodies that come out of this. This is a complex system. So in our view, just sticking the genes in anywhere, as long as they're expressed, is probably not the best way to get to the best antibody. So we're really keen on physiological gene expression. As I said, retain a constant region. And what's interesting is that, as far as I'm aware, every other approach at doing this has deleted some of the mouse genes. Obviously, you don't want to have mouse antibodies as well as human antibodies. I think Kymab is the first company who's actually done this without deleting any genes from the mouse. And what we've done is we've actually silenced the endogenous mouse genes by a very clever way I'm not going to go into too much. But basically what we've done is we've flipped the mouse variable genes in the opposite direction from they're normally transcribed, and put them millions of megabases further away from the constant region and the Ds and Js. Therefore, they can't actually recombine. And the reason we've done that is because there are a lot of genes in the mouse equivalent V genes. There's interspersed genes here, which are important for housekeeping of the mouse. And therefore, we are quite confident that our mouse has all the genes that it needs because it has the same genes it started with. We haven't deleted anything. We're also making a platform because we think diversity is important, and as well as the HKL, which has everything, we actually have ones which have kappa only and lambda only. If you immunize both of those, then you'll only get kappa and you'll only get lambda antibodies. So that gives you a nice finesse for searching for your leads. And I'll mention a few slides in the end in what you call our Supra. Perhaps not the best brand name in the world. It's not a Toyota. But what we're getting at here is really extensively broadening the sampling of our diversity by having multiple strains with differing gene repertoires. So really we think that compared to previous generations, we reproduce and actually enhance. And one of the enhancements, for example, is that for the scientists in the room, the kappa, the distal regions of the kappa light chain in man have an evolutionary inversion, so they're almost never used in antibodies. But we've put it back the other way, just to see if we can get better usage. So that's an example of what we were trying to do with this. And because we built the mouse in a modular fashion, we can do all of those manipulations and continue to evolve. It's not a dead end like a random transgenic. And multiple strains. The knockout strains, because we make these in the ES cells, we can rapidly do knockouts without crossbreeding. And therefore, if you want to immunize and get either use a highly conserved antigen or get cross-reactivity with the mouse, this is a good way to do it. And if you want to do some of the points that were made earlier about rapid readout of biology, because these mice make human antibodies, but they have a background, a genetic background of human antibodies, in the KI we knock in the human target and you've actually got an in vivo model that you can use your antibodies straight into in an early stage. Just to show that they actually work, this is some data from Kymab looking at spleen weight, lymph node cells, spleen cells, and bone marrow cells. And the gold is wild type, and these are two different of our transgenics. And it's very hard to tell the difference. This would not be the case with an Abgenix or a Medarex mouse, which would be between 1 and about 5% of the B cells. We get a robust immune response, and in fact, this case, exceptionally robust. This is 25 mg per mil. And the rest with different antigens, which are different mice and different antigens, which are actually our internal drug targets, so I can't divulge them. And this is a wild-type mouse dosed with cholera toxin, just for comparison. We get all the normal class switching, everything that you would expect in a mouse. I'm not going to show that data. This is an interesting piece of data. This is actually next-gen sequencing on a normal mouse, on two mice, 2 Kymab, and 2 people. And this is looking at the CDR3 length and the distribution of the CDR3 length in non-immunized mice and people. So this shows the normal mouse picture with an average of about 11, 12. The Kymab has a much more human-like pattern, and I think this emphasizes what I'm meaning about diversity. The length of the CD3s in our naive mice goes from about 5 all the way up to 25, much broader diversity than in a mouse, and actually very similar to a human. Data I'm not going to show you, but when you immunize these mice, you actually move this even higher, and this becomes about an average of 17, so we actually get even longer CDR3s. In terms of the proof of the pudding, this is just a few of our antibodies are working on. This is ovalbumin, which obviously doesn't have a functional test, so these are just binders. And I have to emphasize that because we only finished our first usable mouse in September, this is data from a few mice, and the maximum data set here, I believe, is 2 fusions into standard hybridomas. So these are very limited data sets. But with ovalbumin of 100 binders, we get a very nice diversity. This is, I don't know if he's in the audience, but one of our scientists would tell you this is an unresolvable affinity, but he can't tell me what it is. And one of these antigens here is multimeric, so take this perhaps with an avidity effect as well, so we don't actually know the exact affinity. But actually, having said that, with one immunization straight out of the mouse, a couple of fusions, these are quite a not bad set of antibodies, and all these are neutralizing in our assays. We did a primary and secondary screens, confirmatory, etc. So this is quite solid data, but in a very small data set. So we actually think that these mice are doing what they're supposed to be doing and producing high-quality antibodies. Just to finish with the last technical bit is about the Supra. This relates to, if this is 10 to the 14, you've only got 2 times 10 to the 8 B cells. So for a start, even if you took them all, you can't possibly sample it. And if you go to hybridomas, you're leaving 99.9% of this on the table in the mouse. So how can you do better? Well, you can do a lot of mice, is what people tend to do. I mean, someone told me that when he was using Medirex mice, he used to, a single group was 150 mice. So obviously, but we don't seem to have to do that, because in the previous experiment it was like 2 mice or 3 mice. So you do get some improvement if you do multiple mice, but there's an issue, and that is that if you're immunizing with an antigen which has a dominant epitope, you tend to find that you get not only a dominant epitope, but you get actually often a specific V region is overrepresented here. And maybe although this is good for binding, this is maybe what you want pharmacologically. So this is one of the kind of bugbears of immunizing mice, because the mouse doesn't select for functionality, it selects for binding. So what we do here is we thought, well, what if we knock out the V regions that are responsible for a large proportion of these antibodies? Would we get that picture by changing the gene repertoire? And this is in fact what we're working on now, and unfortunately I can't give you the answer to that question yet. So this is still quite theoretical because we don't have the mouse yet, but we're well on the way to having the mouse. But the idea is that we could actually, because the way we make our mice, we make them in a modular fashion, we can actually have one mouse with the proximal V genes, one with the medial, and one with the distal. So we have 3 complete different gene repertoires, but if you immunize all of them, you'll get the full total. And therefore, what we've done is we've developed this idea of having 6 strains which sample different parts of the potential repertoire. So these ones have the different heavy chains, and these are all lambda, and then the different heavy chains, again, subgroups, but all kappa. So you're really covering it from both angles, both in terms of the light chain diversity as well as the heavy chain diversity. So we think this is quite exciting, and it's quite new, and we've only started talking about it now because we've filed our patents. But essentially, we believe a larger proportion of the theoretical repertoire will be sampled. And we've got initial data showing that, we don't have all of these mice yet, but we've got initial data showing that when we compare mice which have different subsets of the IGH repertoire, in other words, intermediate strains, that we do actually get very different usages, as you might expect. And we think this will be very important for epitope coverage. And interestingly, one of the earlier slides I showed you with our first few fusions on a bacterial antigen, we got 4 antibodies which had really good, very good affinity, and they covered 3 different epitopes. So we're actually, we're getting a feel for there's something good going on here. So this is getting to where we are in terms of the Kymab HK, which is the heavy kappa, is actually available, has been available since September. So we've got quite a few of those mice. The heavy lambda will be available at Q2, but we're actually, in terms of our partner use, we're viewing these as one package because they effectively cover all 3 chains. It's quicker, obviously, to make this than it is to cross them all together. The Supra will be available Q2 2014, all 6 of those strains, as well as the HKL, which will be with all of the diversity in one mouse. And then in terms of the target knockout, target knock-in, that's a custom thing. So it's 12 months from whenever we start. Essentially, we will be able to do knockouts and knock-ins available for immunization. And that's really all I wanted to say. In terms of our partnering objectives, because I am, after all, the Chief Business Officer, our partnering requests here are, first of all, we're focused to the company is actually our own drug discovery. But since we have this platform, we are very interested in partnering with pharma. And we are very keen just to actually let pharma companies have the platform internally with no gatekeeping or restrictions. They can do what they want with it as long as they pay us enough money. And on the other side, we're very happy to work with biotechs who have targets of potential interest but don't have an antibody platform. We're very much interested in joint co-development projects to help build our pipeline and the pipeline of our partner. Thank you very much.